Well, first of all, great thanks to Wolf and Jay for this invitation. So just as a brief reminder, the ESCORT trial was a trial in which we tested human embryonic stem cell-derived cardiovascular progenitors in patients with heart failure. And the progenitors were embedded in a fibrin patch that I delivered onto the epicardium of the heart. The study is now completed and successfully met its primary endpoint, which was safety. The first patient I operated on is now 7.5 years from her surgery. And we're now moving to the SECRET-HF trial, which will test exosomes, if I take the title of the presentation, although I think extracellular vesicles might be a more appropriate wording. The objective of the talk is thus to show that indeed there is a logical link between the two studies. Actually, the starting point was a very simple experiment. As I was transplanting the CPCs clinically, we tried to better understand the mechanism of action of these cells, and the paracrine hypothesis was already around, and we designed this very simple experiment where we took the CPCs, the ones that I was using clinically, and cultured them, and then separated the cells in two parts. One part was transplanted conventionally, and the other one was used for collecting the EVs. And we used a mouse model of chronic left ventricular dysfunction, meaning that the injections were made 3 weeks following the indexed infarct. And that was a blinded study, and we used left ventricular and systolic volume as the primary efficacy endpoint, and found exactly similar outcomes regardless of whether cells had been transplanted or EVs from the same cells had been injected. And when we looked at the pathways that had been activated in the recipient hearts, again we found basically similar pathways in both the cell and the EV groups, and more or less all of them contributed to some form of cardiac repair. So then, for a given level of equivalence, the question was why to switch to EVs if they're just doing as well as the cells, not better, but just as well. And so we had to consider a couple of reasons that I would like to discuss now. The first possible reason was that it would be easier to get more reproducible batches using EVs compared with cells. It turned out to be wrong. This shows you the main manufacturing steps during the ESCORT trial, with once the cells had been differentiated from pluripotent stem cells, the phases of expansion, then the sorting, then the inclusion of the cells in the patch. Now, if we consider now the SECRET-HF trial, we still have the same initial steps, exactly the same. We have to differentiate the cells, we have to scale their number up, we have to verify their cardiac specification. Obviously, then we don't have to push the process all the way down as we did. Well, we collect the conditioned medium, which has then to be purified, filtrated, until the fill and finish procedure. Well, this simply means that you still have to go through a series of technical steps which are challenging. They're not more difficult than with the cells, but they are not easier as well. So, once again, using the EVs still requires to work with cells. Now, second reason could be that it's an easier regulatory path. Well, it's wrong. Actually, EVs are not considered as a TMP. They're considered as biological medications, which means that the path may be slightly different, but it doesn't mean that the path is easier. And I just would like to show you the main quality controls we had to do to comply with the requirements of a regulatory agency. So you see here a first series of quality controls, and you recognize that most of them are very similar to what is required when you're dealing with cells. In particular, we had to map the cargo of the EVs to determine the identity of the proteins and the microRNAs, and obviously also do oncogenicity and toxicity studies. And importantly, oncogenicity study, even though the cells are not transplanted, they had to be extensively characterized before we were allowed to use their EVs. A third reason could be that it's easier to adjust dosing, and that might be a better reason. Actually, this is a study dealing with mesenchymal stromal cells and showing that to be therapeutically effective, these cells have to be delivered within a fairly narrow range. Well, all of us who have worked with cells for clinical trials know, in particular when cells are autologous, that it's extremely difficult to end up with consistent numbers. And here you see some data from clinical trials, and you can appreciate the tremendous variability in the number of cells which were delivered. That might be one possible explanation for the failure of many of these clinical trials. Now, with EVs, it's a little bit easier because regardless of the metrics that you will use, once you have fixed the target number of EVs, you can just manipulate and adjust the cell culture conditions until you get the target number. So probably it's easier to end up with a more consistent dosing. Another possible reason is that it might be easier to get potency assays with more straightforward readouts than with cells, and it turns out to be true. I just show you two potency assays that we have been using. You can use them as well with cells, but with EVs, it's probably a little less challenging. The first one is a very conventional cell survival assay where you stress the cells by serum deprivation or now staurosporin and end up with a reduction in cell viability, and then you incubate these cells with increasing concentrations of EVs, and as you can see, there is a dose-dependent improvement in cell survival. So a fairly straightforward readout. The regulators like these kind of potency assays, and once again, you can do that with cells as well, but probably it's a little bit more challenging to get very reproducible dosing. The second one is the conventional scratch assay where you create a wound in a monolayer of endothelial cells, and obviously in a poor media you don't get any healing, whereas with EVs you can end up with a fairly nice healing of the wound. And once again, these kind of results can be made dose dependent. But actually probably the main advantage here, the main advantage for us clinicians has been the lack of immunogenicity, because this is something we have not yet fully addressed yet, but when you use allogenic pluripotent stem cell-derived cells, you need to give immunosuppression, and we know the clinical side effects of immunosuppression. This is the immune profile of the CPCs that we are using. And you see that they do express a little bit of HLA-1 as well as ligands for NK receptors. Now, if you look at the immune profile of the immune profile of the EVs from the same cells, you see that they're basically immunologically neutral. And actually, they do not trigger lymphocyte proliferation, compared here with the control group. And these are 3 different preparations with 3 different doses of EVs, and you see that they fail to induce proliferation of CD4 or CD8. Likewise, they do not trigger NK degranulation. And interestingly, what I show you now is exactly the same kind of data from the clinical grade EVs. And you see that the clinical grade EVs, likewise, do not trigger NK degranulation and do not trigger lymphocyte proliferation. So that might be a major advantage. Now finally, what did we keep from ESCORT and what did we change? Well, what has not changed is the type of cells. In that, we still use cardiovascular progenitors, but what has changed is that we do not longer transplant the cells, but just use them ex vivo as biofactories to produce the secretome. A second change has been that only for practicality reasons, we changed the source of the pluripotent stem cells from ES cells to iPS. That was primarily for reasons of scale-up, ease of procurement, and so on. But we had to check that these were the same cells, and indeed they are. If you look at the CPCs, regardless of they come from ES cells or from our iPS, basically they do express the same markers. So they are cousins or twins, you name them, but basically these are the same kind of cells, and you see here the difference with more major cardiomyocytes. And finally, a major change has been to switch from the epicardial delivery of the cell-loaded patch to IV delivery. Reasons for that was, number one, that because of changes in demographics, it's increasingly difficult to recruit patients who may require an open chest approach. But number two, and more importantly, is that because of the lack of invasiveness of the IV approach, it allows repeated administrations, which are likely necessary to optimize the therapeutic benefit. And indeed, in SECRET-HF trial, patients will receive 3 sequential infusions of EVs. So we did obviously a couple of and several preclinical studies testing this IV approach. I just show you 2 examples. This is a post-chemotherapy model of left ventricular dysfunction. And in this case, it's a mouse model with MRI. And definitely there is an improvement in strain, which, as you know, is a fairly sensitive indicator of left ventricular dysfunction in these kind of models. And this comes along with changes in different markers which go in the right direction. To double check this data, we also did a rat study and basically found similar kind of results, that is, improved functional outcomes here manifest as a reduction in left ventricular and systolic volume. So these data actually are consistent with already reported studies in the literature, as you can see here. And at first glance, this data may look counterintuitive, because if one looks at the biodistribution of intravenously infused EVs, actually most of them end up in spleen, lung, and liver. Well, one possible mechanism of action could be provided with studies which have been done in the field of brain damage. And in a fairly consistent fashion, these studies show that once remotely sequestered in spleen, lung, and liver, the EVs, or the cells, but indeed the EVs, can induce changes in endogenous immune cell populations and tip their phenotype towards a repair pattern. And these cells, when they travel throughout a body and reach the target organ, might actually turn out to be the effectors of the protection. So just to finish, this is the summary now of the manufacturing steps for the SECRET-HF. Collaboration with Fuji CDI allowed us to get clinical-grade cardiovascular progenitors, which were shipped to Paris, and then our GMP platform. We had an additional step following thawing of scale-up, then purification by tangential flow filtration, and finally the fill and finish procedure preceding clinical applications. So once again, I thank you for the invitation and would like to end by really acknowledging those who did the work. This is my dream team, so you see that I am a fairly happy man. And also the people on the JMP platform in Hôpital Saint-Louis who have been responsible for all these translational steps and who have prepared now the batches of the clinical grade EVs, which are just waiting for the final approval of the regulatory agency. Thank you.